Hello, my name is Jaylin, and I am here to teach you a thing or two about mitosis and meiosis. So grab a seat, buckle up, and enjoy the ride.
Now, shall we begin?
The first step to mitosis is this process many know as interphase. Interphase includes three phases, G1, S, and G2. G1 is when the cells grow, the organelles increase, and it produces a ton of protein. S is when the DNA replicates. G2 is when more mitochondria are made, energy is stored, protein continues to be created, and the cell gets ready for division. Prophase is the process in which the nuclear membrane disappears, the centrioles go their separate ways to the opposite poles. Their spindle fibers grow and better themselves so they can live a long and happy life. Sister chromatids all line up on the equator of the cell because that is where the party is at. The spindle fibers find the better half of a sister and attach themselves, so they can take the sisters for a night on the town. All the sisters get in a fight and go with their spindle fibers to the opposite poles. The broken up sisters reach the opposite poles and let themselves calm down and lengthen, so they can be the chromatin they were meant to be all along. The chromatin no longer need the spindle fibers, so they go back home. The chromatin starts to build a nice nuclear membrane to live in. The cells begin to separate and become independent.
You got it, dude.
Damn interphase. Back at it again with preparing the cell for division. The nuclear membrane ditches the party and the freaks come out to chill. The chromatin get a little creeped out by the spindle fibers, so they decide to condense into groups. They all agree to be homologous and come together, forming a squad known as the tetrads. Throughout the next few hours they become very close enough that they decide to trade clothes with each other. In order to understand, you must know what synapsis is. Basically it is when two homologous chromosomes pair prior to segregation, allowing for crossovers. Crossing over is another thing you must know. It is the exchange of genetic material between homologous chromosomes. It occurs during synapsis and increases genetic variation of sperm and egg.
What year is it?
The tetrad squad all line up in the middle of the club. The centromeres align. The chromosomes find some decent spindle fibers to attach to. The tetrad squad then separate to opposite sides of the club, known as segregation. The sisters, however, do not separate yet. The members of the tetrad squad arrive at their respected places at opposite sides of the club. Here, two haploid cells begin to form. At other clubs, such as animal clubs, cytokinesis may occur. New spindle fibers arrive at the club and decide to dance with the tetrad squad. Once again the nuclear membrane that protected them disappears because they are no longer in need of protecting. The tetrad squad all line up at the center of the dance floor once again. The spindle fibers find the sister they are most compatible with. The tetrad squad says their goodbyes and go up to dance with their new spindle fibers on opposite sides of the club. The cytoplasm starts to pinch off and form two more dance clubs because of how popular they turned out.
Well, good day to you. And I will, we're done. Just, squeeze by you here.